Right, well thank you very much for the invitation, and I think actually you've given me quite a tough slot. It's straight after lunch, so you're probably feeling a bit sleepy, and also I'm after Chris Fagan, as you saw, is a bit of a tour de force. So I'd like to start my talk, I hope today, to teach you a little bit about the immune system, at least from my point of view as someone who's been working on T cells, which are a part of the immune system. I've been working on T cells for over 20 years, and also teach you how say a little bit about how CLL influences the immune system and what we can do sort of going forward to look at the interaction between the two things. So I'd like to start my talk by really acknowledging some of the people in the lab who've helped me with the research. I've had a whole series of PhD students, basically all funded by Cancer Research Wales to help me with the research. And I couldn't have done any of the research without Chris Fagan, obviously, who comes over twice a week with patient samples, and also Chris Pepper, who's been a big influence on the scientific side. So I'll leave that there and go on to the next slide. And I've been asked to talk about the immune system. Now, you saw some of Chris's slides were quite complicated, so I like to say the immune system is much simpler than that, and I like to illustrate that by showing this slide. Now, this slide, you've probably worked out, is a map of the Tokyo Underground system, Tokyo Metro. And I can assure you that I've been lost on this metro as well as many others. And really, it's to illustrate how complicated the immune system is. It's composed of many components, and they all feed through, and the whole organism somehow keeps going. So even if one of these lines in Tokyo, oops, how do I operate the printer? Is that, sorry. Pointer. Doesn't matter. Even if some of these lines break down, the others still carry on working. And so the whole city keeps on moving. So the immune system is very complicated, lots of parts to it, and some parts don't always work. And actually the parts that don't work, more parts don't work as you get older. So now the thing about CLL is, we know that there are some parts which don't work. But actually, many patients with CLL, they don't seem to have any obvious immune problems. So it's quite interesting to find out why that is. So the immune system, as I mentioned, is a system. So we've got to think about organs, lymphoid organs, and I'm sure you'll know about lymphoid organs, your lymph nodes, bone marrow and spleen, but also what we're focused on, the cells in the immune system, and particularly the T and the B lymphocytes, and obviously the B lymphocytes are the CLL cells, which are, as we found out from Chris earlier, accumulating in your blood and causing that CLL disease. And there's other things that involve the immune system. Lots of things are secreted by these cells. So there's, as well as the cells moving around, protecting you, there are things which are secreted by the cells, which are in your blood, which give you defense against infection primarily. So, the essential difference between B and T cells. So B cells, these are the CLL cells. These are the ones which are growing and accumulating in your blood. And generally their job in normal health is to attack invaders outside the cells. And they do that by making antibodies. So the antibodies float around in your blood and they latch onto bacteria. T cells attack things which are really inside cells. And I'll talk more about that in a minute. So their job is to look at things inside cells. And both of them really act to protect the the body against infection, really. So here are B cells, and as I mentioned, their job is to make these antibodies, these soluble molecules which are in your blood, latch onto bacteria. And of course, in CLL, these cells are abnormal, so you might not get the full range of antibodies that you need to fight all infections, and this is something that Chris talked about earlier. But really the focus today is about T cells. They're more engaged in sort of hand-to-hand combat. And their job really is to look at things that are lurking inside cells. So that might be viruses, it might be malarial parasites, it can be bacteria like listeria. So all sorts of things lurk inside cells and can cause disease, and T cells detect those and eliminate those. So, as I mentioned, we get a lot of information from studying the blood samples that Chris gives us, and as you can see here, this gives you a sort of snapshot of what's going on in CLL. So I talked about B cells and T cells, and of course the orangey looking cells are the CLL cells, which outnumber the T cells. Now, in a patient in someone who hasn't got CLL, it's the other way round. The blue cells are far more frequent than the orangey cells. So there's a reverse in CLL, and that has a dampening down effect on these T cells. I mean, for me, as an immunologist, CLL is quite a good model for cancer immunology in general, because in the same tube, I get cells which are from a cancer, and I also get the immune cells, and I can look at the interaction between the two of them. For other diseases, it's very difficult. For many years, I worked on prostate cancer, and it was almost impossible to get a piece of cancer and the blood sample from the same patient. But here in CLL, we've got the same cells, the two different cells in the same tube. So as I mentioned, CLL cells are a part of the immune system, and there's a lot of suppression of the immune system in CLL. That results in more infection in patients with CLL. The tumour cell itself has been shown in all sorts of tests to be quite suppressive for T cells. So if you mix them together, T cells which are activated might become resting or stop proliferating. So the tumour cell itself is suppressive to T cells. And then over the years, many T cell abnormalities have been reported. So that's something I'm going to come on to, and that's one of the main focuses of our research. The interesting thing about CLL, it's a cancer growing inside the body, but there doesn't seem to be any apparent immune response against it, which is quite odd. And that probably is in part due to the fact that the tumour cell is very good at suppressing the T cells. Now, in terms of doing research on CLL, we're really focused on the two main areas, and these are the two main areas that Chris talked about earlier, and that is looking at patients, the watch and wait group, if you like, which have no symptoms. So can we look at the immune system and develop some idea of whether changes in the immune system can predict whether patients will need treatment or not? And then, of course, the other important group are the patients who do get progressive disease and have conventional therapy, and what happens to them. So some of the therapies Chris talked about are extremely promising, but there's a whole branch of research going into immunotherapy. And immunotherapy has been spread across cancer, really, as, if you like, the next big thing. And the reason it's the next big thing is I'm going to come on to in the next few slides. But again, this group, can we monitor the immune system and maybe predict patients who will respond well to therapies or don't respond so well to therapies? And can we use knowledge about the immune system to group patients who might benefit from particular treatments? So I think you all know why we need prognostic markers, so I'm not going to talk too much more about that. But the new therapies for cancer, and again, this is sort of across the board for all cancers. We know that conventional treatments don't always work, and we've heard about some of the toxic side effects today. And the reason why immunotherapy is really being sold as the next best thing is it's a biological therapy, so you use cells from the patient. And the thing about the immune system is it's very specific. So the idea is that you can target cancer cells alone and then leave the healthy tissue untouched. And that's something that chemotherapy and radiotherapy don't do. And so as a consequence of that, you should really get no side effects from immunotherapy. But again, Chris mentioned there are side effects from new immunotherapies, and it's a question of weighing up the benefit of the therapy versus the side effects. But anyway, that's really why immunotherapy has taken off, or research into it has taken off, is because of this idea that you can have therapy or potentially cure with very few side effects. Now, immunotherapy involves more than just going to Boots and buying a box of tablets. So before that, I talk more about immunotherapy. I'm really going to take a step back and look at what do T cells do. So I'm not sure if everybody in the room understands what T cells do in a normal immune response. So if you look at T cells under a microscope, they're very boring cells. And in fact, in most of you, if I took a blood sample from you, your T cells would not be doing anything. They'll be like these two chaps here, just sitting around, not doing anything. And that is actually the normal state for the immune system. It only fires up into action when you have an infection. That's the idea. If it's on all the time, you'll probably start attacking your own tissue. And that's a bad thing. So it's sort of controlled aggression, if you like. You only get it when you have an infection. So why are T cells important? Well, the best way to understand whether something's important is to find out what happens when it's not there. And so there's a disease called SCID. And some of you will know the song by Paul Simon, The Boy in the Bubble, and some of you might even know the film with John Travolta about the boy in the bubble. Now, the boy in the bubble was a boy called David Vetter, who only lived 13 years, and he had a disease called SCID, or Severe Combined Immunodeficiency Disease, which is quite a rare disease. About 1 in 100,000 people have this disease, and this disease is caused by a mutation, and it stops the development of B and T cells. So basically, if you have SCID, you have no immune system. So when David Vetter was born, the clinicians decided to put him into a big bubble to protect him from the outside world and all potential infections. So he lived in that bubble, and basically he was part of a long-running scientific experiment. So the researchers convinced his parents that they would be finding a cure for him while he was in this bubble, doing research. Now, in 13 years, they didn't find a cure for him, and they decided then that they would give him a bone marrow transplant from his sister. And unfortunately for them, they didn't know that his sister was harboring a virus called Epstein-Barr virus. Now, most people in this room have Epstein-Barr virus, and for most people, it doesn't cause you any ill health. But because he had no immune system, when they put bone marrow from his sister into him, he developed lymphoma caused by the virus, and he died 2 weeks later. So really, this example is to illustrate how important the immune system is. He had no B or T cells, and he had to stay in a bubble. Occasionally, he was let out in this NASA spacesuit. But the other thing I want to illustrate is that his sister had this virus, which could potentially cause lymphomas, and she was dealing with it fine. And again, it's the same virus that most of us in this room have got. So the immune system can keep things under control before they get to the cancerous stage. So it's very important. The reason why T cells have been mooted as the next big thing for immunotherapy is based on studies like this. And this really shows how powerful the immune system can be. And I apologize, it's a rather gory picture after lunch. But this shows an American man with melanoma, which is spread to an area under his arm, and he's got this solid lump under his arm, big, bulky, vascularized tumor, and obviously he has resisted all conventional therapies, including surgery, chemotherapy, etc. And what they did with this particular patient, and a whole group of patients in America, was they cut a piece of the tumor and they extracted all the T cells from it. And then they grew those T cells to billions in number, and then they infused them back in him. So that's a technique known as adoptive cell therapy. So CAR T cells are a variation, if you like, of adoptive cell therapy. You're putting cells in to a patient, usually their own cells. And you can see here the results are miraculous for this particular patient. And it doesn't happen for every patient with melanoma, but you can get these amazing responses showing you how powerful the T cells are. So T cells can destroy something big and bulky like that. Now, closer to home, this is the report of an 8-year-old girl from Manchester. She had a brain tumor. So I'm not a medical doctor, but even I can see that in this CT scan that there's a part of the brain that's sort of lighting up white. So that's a brain tumor caused by Epstein-Barr virus. And she was in a coma, and they, on compassionate grounds, they asked a lab in Edinburgh to send down T cells which were against Epstein-Barr virus. So these T cells were taken from a healthy donor. They recognized the virus, and they'd frozen them down. So they then infused these cells into this girl, and you can see after a couple of infusions, the lesions have disappeared. So that was back in 2005. I think she said some other treatments, not immunotherapy, and then apparently she's leading a healthy life at school. So what I want to show from this is that T cells, as well as being very powerful, destroying this big lump of a tumor, are also very sensitive. So these T cells have crawled into the brain, destroyed the cancer, and apparently gone out again without doing any harm. Now, you think about it, the brain is a very delicate organ, and one of the reasons why meningitis is such a dangerous disease is because it involves inflammation of the brain. So the brain is one of the organs you need to protect to keep on functioning well. So in this case, T cells have gone in, destroyed the cancer cells, and come out again. So very specific. So this power and this specificity are the things that really drive immunotherapy. How does this work? So we've got a clue on how this works. So there are T cells which are called CD8 T cells or killer T cells. And I'm just going to show you there's two here. So this looks a bit like the Cookie Monster, but these two are cells stuck onto a big cancer cell. And I hope, if this works, to show you a video which a medical student made in my lab of these killer T cells in action. Okay. There we are. So you can see there's a bit of wrestling going on at the moment. The two cells on the top, the two little cells are stuck, and they're wrestling with the big cell in the middle. That's the cancer cell. And after a while, you'll see that those cells deliver what's called the kiss of death. And now you can see that that cell is bubbling up. It's bubbling up through this process called apoptosis, which Chris described earlier. And this process of apoptosis, I'll just show you the video again so you can see it. This process of apoptosis means the cell is dead, it's dying. And so there's all this wrestling going on, and the reason we hadn't discovered this before was because when we took this time-lapse video, it took about 7 hours for it to happen. So these cells are very resistant. So the beauty of that whole process is that those two cells which were stuck onto the cancer cell can go off and do the same thing to other cancer cells. So if you like, they're serial killers. Now, at this stage, you're probably asking, that's fantastic. You've shown that example in melanoma, the skin cancer, and you've shown the 8-year-old girl, and you've shown that T cells are killing cells in the test tube. Why can't we do this for every cancer? Well, there are 3 good reasons why you can't do it for every cancer. The first is that targets. You need to be able to push the immune system onto cells which are cancerous, but leave the normal ones untouched. Second is numbers. When you try and find T cells which recognize cancer naturally in the blood, they're very rare. So you need to boost numbers. And last but not least, cancers are proliferating and growing, and they mutate their own DNA, and they can figure out ways of escaping the immune system. So you've got to think about defense as well and try and outsmart the cancer's defense mechanisms. So in terms of the first challenge, I like to use this example. So this is the Amazon warehouse in Swansea. And you can see there, if you look carefully, you can see somebody walking around there. So how do they know which box to put in which hopper? They know which box to put in which hopper because they've got a handheld scanner and they can read barcodes. So that's a bit like what T cells do. They circulate around the body. They've got a scanner, which is a receptor on their surface, and they look for things which are different. So if we can identify what those things which are different are, the targets, and make the barcodes, maybe we can train the T cells to recognize things with the right barcode. Now, finding those T cells is a bit difficult. I liken it to panning for gold. So we've looked at CLL blood, and we've looked at particular T cells which might be useful, and we found there are about 1 in 100,000. So it takes a lot of work to get those T cells out of the blood. So could we boost their numbers? Well, we could boost their numbers by using vaccines. Now, these are not quite the same as the vaccines you use for flu or hepatitis. These are vaccines designed to boost the numbers of T cells. So these are vaccines to treat a disease, not to prevent it. And the other way you can do it is to boost the numbers outside the body with this technique called adoptive cell therapy, which is what the CAR T cell therapy is based on. So here you take blood or a piece of tumor from a patient, you grow out the T cells to huge numbers, and then you infuse them back in. Defense is very important as well. So there's many strands of research which come together to understand how we can best develop immunotherapy. So this is a picture of Bow Maris Castle in Anglesey. And really it's to show you that at first it seems like an impregnable obstacle, but actually if you can look at it from a different angle and look at some of the structures within it, we might be able to find a weakness. So there's a whole strand of research which is also looking at understanding the defense mechanisms that cancers have against the immune system. So it's marrying all these together which will help us develop immunotherapies. And this is the sort of research we're doing in my lab. So we'd obviously like to develop new treatments for leukemia using T cells, based on looking for new targets and understanding defense. And another strand of our research is trying to predict which patients will need treatment in the watch and worry group. And so generally what happens is, this is just to show what happens when we do lab research. Chris comes over with samples and we separate out the lymphocytes, and then we can freeze them in liquid nitrogen, or we can use a device called a flow cytometer to look at them, or we can grow the cells. And I'll come on to what we do with the cells in a bit. Now, T-cell research, I've been working on T-cells for over 20 years, and when I was doing my PhD, life was quite simple. There was really only two types of T-cell. Now, I've talked about these killer T-cells, these CD8 T-cells, and the CD4 T-cells. CD4 T-cells are very important. They're the cells that disappear in HIV infection. And one of the consequences of HIV infection is that you lose your immune response. So CD4 T-cells are also important. But nearly all the focus for immunotherapy has been on these killer cells. And you can probably see why. They have a direct action on the cancer cell. Now, the problem is now we have an issue where there are many different types of T-cells. So we used to have just CD4 and CD8, and now there are all these different varieties. So how do we work out which T-cell is doing what? And we can do that using this device, which is a flow cytometer. And what this does is it allows us to look at different tags on the surface of the cell. So just like you can spot someone that you know in a crowd based on their hair color, the way they walk, or their eye color, we can spot what types of cells we have in a sample by the molecules on their surface. And we can do this with fluorescent tags. I'm not going to go into detail of how we do it, but we can separate out all the cells based on this sort of identification kit. Another complication is the T cells in the blood develop with time. So they might develop with age, and they also develop with infection. So you don't have T cells which are floating around which stay the same. They actually mature. So this gives you an example of how they mature. So you start off with T cells which have never seen anything, a naive T cell. And the naive T cell might see a virus. The naive T cell will expand and proliferate, and it will develop memory. So next time it sees a virus, it will react very quickly. But as that memory cell develops with age, it gets older and older, and it actually starts to lose its ability to do what it used to do when it was young, like all of us. And so we end up in a situation where you have someone like Keith here, who's lived a good life, and you might be at the state of exhaustion, which is where you lose your function, or senescence, where you just fail to proliferate anymore. So you have a mixture of all these T cells floating around in your body, and in healthy people, there's a good mixture of these cells. So you've got quite a few naive T cells to help you respond to new things, and you've got some cells which remember past infections. So this is the sort of results that we generate in the lab, and you're probably thinking, what the hell is he going to talk about now? I'm already confused. So I'm going to simplify it a bit and show you some pies, because people like pies. And all you need to focus on here are the colors. So look at the purple and look at the blue. On the left, you have CLL patients that we've studied. So in this particular case, there's about 40 patients there and about 20 healthy donors, maybe 14 healthy donors. Now, I talked about these naive cells. So these are the young cells. They've never seen anything. So you see in the healthy donors, they're a good percentage, but they decrease by quite a large amount in the CLL patients. And then the cells, which are purple, these are memory cells, which are kind of the Keith Richards cells right at the end there. There's many more of these in CLL patients. So in CLL patients, you're being sort of hit from both sides. You're losing the young cells, and then you get more of the old cells. I can probably sum it up even easier using this analogy. So you have the healthy donors on the right. So you have more young cells in the healthy donors. and fewer of the old cells. And then in CLL, you're losing your young cells, and you're getting more of the Keith Richard type, which are not so functional. Now, at this point, you're probably saying, well, that's all very well. You've done all this fancy stuff, machines and different colored things, and you've shown all these T cells. You know, what difference does it make to a CLL patient? So you might be asking, where's the beef? And at the threat of being sued, I can say you'd probably be unlikely to find any in this product. Right. Well, the beef is that actually this, by grouping the patients into those which have a lot of the memory cells, so they have more CD8 cells with those exhausted phenotype and fewer of the young cells, there seems to be a difference in how their disease progression. So I'm not going to go into the details of this slide, but you can see these patients do a lot better than these, and these are the patients who have the most cells, which have that sort of Keith Richard look about them. So it seems that this particular set of T cells can predict whether a patient's going to need treatment or not. So this graph is all about treatment and whether patients have disease progression. So the other question you might ask is, CLL patients have got more of a certain type of T cell, right? And usually you'd think more is better, particularly more of the killer type of T cells. So this is what you want, lots of these killer cells patrolling around, potentially killing CLL cells. But actually what you have is this. You have T cells apparently not doing anything, and maybe T cells which have been corrupted. We still don't know why there is that correlation between more of these particular types of cells and the disease getting worse. And so what we're working on now is whether or not those T cells, clearly in patients who have a progressive CLL disease, they have more CLL cells in their blood. So is it because there are spectators throwing fuel on the fire, or is it because there's a fire and the spectators are coming to watch? We don't know. So we're trying to work out what those T cells do. What this really does, in summary, is echo the words of a great American leader. So in this case, actually, having fewer of these cells is better. The patients with more of those types of cells actually have poorer prognosis. And I know that we've laughed at American presence in the past, figures of fun, but actually we probably look back on this slide and think of that these are the golden days compared to what's happening now. So we're continuing our research, because obviously you saw that we only looked at, I think, 60-odd patients in that graph, with 6 in the other group. So we'd like to open up. So no one's going to believe us because we've studied less than 100 patients, so we're going to do more patients. But now we're thinking that maybe we can use these sort of tests where we look at T cells and what type of T cell there is in the blood to group patients with treatment. And actually, as an immunologist interested in the whole body and responses against infection, I'm wondering if we can use it as a kind of index of immune health, because we know that CLL patients as a group don't respond so well to infection and vaccination as healthy individuals of the same age. So one thing that Chris and I would quite like to do in Cardiff is to do a vaccine study, looking at the partners and patients, looking at cells in the blood and whether we can group them, the patients that respond to vaccine or not, depending on the types of cells they have in their blood, T cells they have in their blood. And of course, what we haven't done in the last few years is do this, find a way of developing killer T cells for CLL. Now, Chris told you about the CAR T cells. Obviously, there are a lot of people working on this. Chris told you about CAR T cells, and this is where you give every T cell in the body the ability to latch onto a cancer cell, in this case CLL. And you do this by taking some cells from a patient, and then you infect them with a virus. And it's the virus which has a molecule in it which helps it latch onto the CLL cells. And then the patient basically gets an infusion like this of cells that have been grown in the laboratory. And as Chris mentioned, actually, CAR T cells have been approved for clinical use in America. The first trials were done in CLL, but actually it's not approved for use in CLL. But he mentioned the cost, which is $475,000. I'm not sure what that is in British pounds, probably about 360,000, I think. So expensive. And it's not escaped the attention of drug companies, as Chris pointed out, that this might be a lucrative business. So there's lots of companies involved in developing these CAR T-cell therapies, because not just for CLL, but it could be for other cancers. So a big market. But it's not quite as straightforward. And Chris alluded to this. There's a big cost to growing these cells. The companies argue that there was a big development cost, and so that has to be recouped for the price of the product. But a lot of the cost comes because growing the cells is quite labor intensive. You need very high levels of sterility, because the only thing you want to put into a CLL patient are the cells which are targeting the cancer. No bacteria, no fungus, nothing, just the CLL cells. And then there have been some side effects, fatal side effects, and these don't often get reported. So in this trial, in a trial run by Juno Therapeutics with CAR T cells, 6 patients died from chronic inflammation of the brain. So this was a receptor designed to target leukemia cells. And what happened was a lot of angry T cells ended up in the brain, which was fatal for those patients. And that couldn't be treated with steroids or any anti-inflammatory. So that product has since been withdrawn. But we don't really know why T cells which are targeted against a leukemia might end up in the brain. So that's a bit of a mystery. And so there's always that, if you pardon the pun, in the back of your mind when you're thinking about CAR T cell therapies. And last but not least is safety. How do we get the CAR into a T cell? You do it with a retrovirus. So you use a recombinant virus to infect lots of cells and you put them into the body. So there are still some issues with using these viruses to arm the T cells. Now, CAR T cells are still being developed, but there are other potential therapies based on T cells. So we investigated an agent in the lab. So this agent is an antibody. So it's been engineered so that it glues together T cells and tumor cells. And you can see in this picture a CLL cell in red surrounded by half a dozen T cells. So when we take some cells from a patient, put it into a test tube and add this antibody, we find that all the CLL cells die. They get killed by the T cells, which are clustering around. So it's forced them together. So that was very promising in the lab, and I'm not sure what the state of this agent is in CLL. I don't think it is being used in CLL at the moment. So that's one way of using T cells. An alternative way of using T cells is by basically forcing them together with a sort of glue. And then the other way is to perhaps use a vaccine. I talked about vaccines before. This is not a conventional vaccine. This is basically trying to boost the numbers of T cells in your blood. And you can do this with synthetic peptides. So little protein fragments you make in a lab, and you inject those, and you hope that you can boost the T cells, and then the T cells will go and kill the cancer cells. Traditionally, this has not had much success compared to CAR T cells and adoptive cell therapy in general. But there are companies working on it, primarily because you may be able to combine therapies. So instead of trying to use this approach on patients who've got lots of CLL cells in the blood, maybe it might be more effective if the patients have had a prior treatment. So if you like, you lower the load and you get a chance for the T cells to respond. So conclusions. I hope I've convinced you that studying T cells may be useful for prognosis, perhaps. I think as an immunologist for assessing immune health, and we can look at a whole range of things to do with immune health. Maybe we can use them clinically to match patients to new therapies. Perhaps we can use them to understand the response to new therapies. What happens to the T cells in a patient that's been treated with ibrutinib, for instance? And what we saw, which I didn't talk about, was that if we look at the immune profile of CLL patients, and the average age of the CLL patients in our cohort was about 65, when we look at that profile, and we look at similar profiles from studies done in Scandinavia of healthy populations, they have an immune profile of someone in their 90s. So although our patients are 65, their profile looks like they're in their 90s. So the disease has aged the immune system, and it'd be quite interesting to understand how that's happening and whether we can do something about it. And the last part of that talk, I talked about the new therapies, which have enormous potential. They are still experimental for CLL. They remain expensive. Some of the companies have argued that they will get cheaper from economies of scale. So if you have more factories making T cells, you can make it cheaper. They do have side effects, and actually the side effects can be unpredictable. But they continue to be developed, and Chris showed you lots of adding on different things to try and make them better, sort of like a Lego set of adding on things to try and make them bigger and better. So actually, before I go on to that, I talked about Keith Richard earlier, so I just want to sort of compare and contrast two members of the Rolling Stones. So Keith Richard obviously has lived a full and active life and enjoyed himself to the full, but he's still going strong. Mick Jagger, by contrast, has been very fit and kept himself in very good shape, and he's still going. So I think really the conclusion I'd like to leave you with is that it really doesn't matter in some cases that the things we're trying to do to predict which patient might do better or respond better is largely determined by your individual characteristics. And so Keith and Mick have led different lives, but they still got the same result. They're still on stage playing rock and roll. So my concluding statement is, go and enjoy life and see what it brings. Thank you.